Dun, dun. Today on Science America, prion protein misfolding and the devastating effects these supposed monsters are having on millions of victims. Hi Kathy, Raquel G here reporting from EBSCO Research Facility. Today we're going to be talking about prion proteins, better known as PRP, that are usually found embedded within nerve cell membranes attached by a lipid anchor, shown in yellow at the bottom of this picture. In nature, prions are found in two different forms, a normal conformation, titled here PRPC, and misfolded forms, labeled here as PRPSC. As we will see later, misfolded prions are the ones that can cause havoc within an individual, causing fatal neurodegenerative diseases. Normally, PRPC is largely made up of alpha helices, 3 to be exact. It also has two beta strands, one N-terminal and one C-terminal. The NPSA calculation indicated that random coils, extended strands, and alpha helixes make up the majority of the protein's normal secondary structure. As a result, this conformation allows the PRPC to be susceptible to protease activity, which is a regulatory measure. Various protein engines have indicated that the cytoplasm is the probable location for the protein, which makes sense due to the proposed transmembrane interactions PRP is associated with. The 253 amino acid long chain has a theoretical pi of 9.13, which is pretty basic, and a molecular weight of 27,629.1. But the only way to be sure of these values is to run the protein on a gel against molecular weight standards or isoelectric point standards. Most data indicates that the actual pi is much lower in the brain, and this is because of N-linked glycans containing 4 terminal sialic acids as shown below. On another note, the chart below shows protein sequence similarities within families. Unfortunately, there were no predicted family relationships, although the only thing worth noting here is that the G0051260 protein homooligomerization and cellular component membrane came up as a similarity. Earlier we used a properly folded bird to represent a normal functioning PRPC prion protein. Some of what goes into proper functioning is the relationships and interactions the PRPC has with other molecules. This chart outlines an extensive list of normal cytoplasmic interactions, including GRB2, which is a signal transduction molecule located in the cytoplasm, TREC1, which is a transmembrane molecule regulating potassium channels, and NCAM, which is a cell adhesion molecule also located transmembrane, all showing that prion interactions are involved in essential cellular functions. Normal functioning prion proteins also play a role in preventative measures, such as protecting against apoptotic and oxidative stresses, which could lead to impaired mitochondrial function, defects in ubiquitin proteasome systems, and the presence of aggregated proteins, changes in iron metabolism, and inflammation. They also act as a maintenance crew for synapses and work towards adhesion to extracellular matrices and transmembrane signaling. Understanding basic prion protein structure and function has allowed us to understand the prion's importance within the body. But what would happen if the structures of these proteins changed? If you remember our properly folded origami bird, you'd realize that he's not looking so hot here. And as you recall, we named him PRPSC for the misfolded protein. Protein misfolding can happen for many reasons. It can happen spontaneously, or when changes in pH occur, such as shown here, or they can be inherited. When a protein is misfolded, there are many obvious structural differences. For example, the secondary structure of the PRPSC prion has many more beta sheets, unlike before. where we can compare them as 45% are now beta sheets to 3% in the PRPC, normally folded prion. This is a problem because they cannot be chopped up as easily by proteases. Another problem is that no tertiary structures of the PRPSC can be detected because of the amount of aggregation built up. As we all know now, structure always influences function. How will this change in the way that the prion is shaped changes function? The long fibrils that are created are unable to be chopped up, begin clogging the brain, where no normal functioning can occur after a certain point. Even though prions are not viruses, it looks like the prions are luring other prions to turn to the dark side and to act inappropriately. More specifically, at the sticky N and C terminals, the highly organized proteins recruit new prions where they can take the same configuration as the PRP-SC protein, each new subunit taking on the properties of its original. As this process repeatedly occurs, amyloid plaques form, as shown in blue, and causes eventual cell death. As this process repeatedly occurs, we will see extreme effects in humans and in other animals, such as mad cow disease, CDJ disease, or scrappy disease in sheep. Humans can obtain these diseases by eating the misfolded protein in bad meat, during general surgeries where medical equipment has been exposed to the misfolded protein and used repeatedly. This can also happen spontaneously in rare events, as well as be passed on and inherited in grave situations. Unfortunately, these proteins cannot be destroyed by conventional sterilization procedures, and health guidelines need to be increased in order to reduce the devastating effects of misfolded prion proteins. That's all we have for Science America today. Make sure to tune in tomorrow for more interesting protein facts. Thank you for watching.